Yeah, so I'm very happy to be here. Thanks a lot for the invitation to this interesting meeting. So I'll speak today about some ideas we had and we are working on for quite a while about the link between 3D structure of chromosomes and function. So this is a rather big question in the field, and essentially what we are going to tell you is a perspective from a modeler. Although we work with lots of collaborators from experiment, so which do contribute data, but I will discuss model simulation. So it's Peter Cook from Oxford, Nick Gilbert from Edinburgh, Christoph Antonis. This is work mainly done for the hardware model, at least by Michael Chang and Claes Battaglia. The toy model that I will present for the first two threads is done by Davide Marenduzzo, Chris Rackley, myself, and Carolina Pereira. We did a simulation for that. Giuseppe Negra and Max Semeraro are also building, taking forward this model. And the idea that we discuss about the structures and how they link to functions. First in a toy model, and then we'll try to go on to a real situation. So, you're probably very familiar, but if you're thinking about chromosome organization, so this is a multi-scale problem where you have DNA, which stands up as a right-handed double helix, is wrapped around some proteins called histones, and we have a packing problem because there are about 2 meters of DNA in a single cell. Okay, so there is a huge problem in packing, and the wrapping of the DNA and histone is the first level. We'll be speaking about further compaction, which is certainly needed, but is a little bit less characterized experimentally. But so chromatin is a polymeric material which is made up by DNA and histone proteins, and that's what I will use interchangeably with DNA. So it's DNA in a human or eukaryotic cells. So the first problem that I'll discuss is this structure, and it's very much linked to phase separation and microphase separation in the cell. And this comes from the observation, which was done probably 100 years ago first, that the proteins in the cell are not in the nucleus, are not uniform, but they form often clusters. So examples of clusters are Cahal bodies, where we found many times ago. Polycomb bodies are found in the fruit fly and are associated with gene inactivation. PML bodies, pro-myelocytic bodies, are these ones here, and they are associated with PML proteins, and the lack of PML bodies is observed in some disease such as cancer. So you see, I've already named 3. Usually these proteins form clusters by themselves, and this is an example of microphase separation in the nucleus. I should check here, actually. Sorry. So microphase separation here means arrested phase separation, or the phase separation doesn't go to completion, but you end up with clusters of self-limiting size. Okay, so this is commonplace in this field. So another example of microphase separation here is that of transcription factories. So I will tell you that there is sometimes an extra line, so I'll tell you about it. So transcription factories were discovered in the 90s, and there what you see green, so red is the nucleus of the eukaryotic cell, so that's a HeLa cell, human cell. And what you see green is the bit of RNA which is being made, so that's called a nascent RNA, it's dyed in green. So it's associated with the polymerases, which is making it. And you see that it's not uniform, but it's again in clusters. So it's a fourth example, it's another example, there are very many, of these intranuclear clusters, protein clusters. These are also often called nuclear bodies. Their size is between 100 nanometer and 2 microns, so very much in the colloidal regime. So the transcription factor is, what is written here, is that it forms with 3 types of polymerases. There are 3 types of polymerase in eukaryotic cells, and an interesting fact is that this polymerase is specialized from different factories. So the Pol1 forms 1 factories associated with the nucleolus, if you're interested. Pol2 forms another type of factories, and Pol3 another one. Okay, Pol3 is rare, but they're different. So what we want to find, now what I will discuss, is that the typical model to understand phase separation is to have protein-protein interaction. There's nothing wrong with that. I will show you, though, a different perspective, which is based on the fact that all these clusters are associated with DNA. So let's start with the toy model and see what happens when we consider many protein-binding protein or DNA-binding protein. And we consider, so these are the red things here. They have interaction with the DNA of chromatin, which is blue. And let me also say that there are two types of interaction. So I will give you an intermediate model because it's slightly more interested. This is more motivated experimentally. So there is a low non-specific interaction for blue, okay, in this case. So the red sticks to the blue, and that's perhaps due to electrostatics, okay? So usually the proteins, if you want to bind to the DNA, you'd rather have a different charge, so it will be positive, and the DNA is negative. So that is already a first step. And then you've got specific interaction, which typically depend on the sequence. So you've got specific and non-specific low affinity and specific high affinity interaction. And this is again typical for most DNA binding proteins. A key assumption in this model is that these things here are multivalent. Okay, so this is the crucial for the results. So you have multiple binding sites. So this would be the natural case if this sphere is a complex, as often is the case. So if you've got a polymerase and a couple associated transcription factors, you will have multiple binding sites. Okay, so that's the model. The red guys stick to the chromatin, and then they stick strongly here, weakly here. No interaction between the red spheres in this model. So what is shown here is the result from a computer simulation. And you see that multivalent binding leads to clustering of finite size. So it leads to microphase separation. Here we have removed the chromatin just for simplicity of visualization. So we call bridging-induced phase separation after different names, BIPS, so which is like mix, but with a B. So we changed the name recently. So it's a microphase separation in this case. For the microphase separation, it's crucial that you have this non-specific binding and the specific binding. And I'm going to say that this is a Tony model, but this is recapitulating at least the framework. So it basically the main results that you find in experiments. So for instance, in this case, the appearance of transcription factors. So you could imagine that the proteins are complexes, as I said, of transcription factors and polymerases, and they would make these clusters, which is very much like those factories there. So why do they come about? So this is a cartoon by Michael, which is a bit better than the one that we do. So the idea is that there is a very simple thermodynamic in this case even. We can have the model of equilibrium, but in this case, this can be understood thermodynamically, positive feedback. So you've got the proteins which are multivalent, so they've got some sticky sites that are specific here, and they will increase, because they are multivalent, they will increase the concentration of chromatin, but chromatin contains both non-specific and specific binding sites. So if you increase the concentration of chromatin, you will also increase the concentration of binding sites, and you will recruit further proteins from the soluble pool. Okay? So now you form cluster this way, the clusters will grow, it will not grow to infinity because the fact that you have this specific binding creates loops, which you might have seen before. I might show you again the movie. So you can see some looping here, which should be just okay, hopefully. And these loops create an entropic barrier, essentially. I can tell you a bit more if you want, both a kinetic and an entropic barrier for cluster merging. Okay, so that's why it's microphase separation. See the loops there very clearly. So this is bridging-induced phase separation, and in this case, it leads to microphase separation. So, as we said, we have the transcription factories experiment. The other interesting thing is that in this case, there is no interaction between the red proteins. If you have now two types of proteins, the red which sticks to the pink beads and the green which sticks to the line beads, so they have specific interaction for separate beads, but non-specific for the rest, you can form specialized clusters like polymerase 1, polymerase 2, polymerase 3. Whereas if you want to explain with the other model that you could have, which is nothing wrong with that, but in this case, you would have to have protein-protein interaction, which is like a matrix, so that red interacts with red, green interacts with green. So if you have multiple proteins, this starts to become difficult. Whereas in this case, there is no interaction. There is coming only through the DNA, the clustering. So this bridging-induced phase separation also gives you a simple mechanism to create A clusters and B specialized factories. So I've already discussed the mechanism is increased concentration. So the binding, because it's multivalent, it increases the concentration of chromatin, and this sucks further proteins from the soluble pool. It can work also between for different colors, so that's how you get the specialized clusters. Okay, so all this is in theory. It's also nice to say that there is some experimental development going on, and for instance, Kiesdecker lab has shown that cohesin can work in vitro. Cohesin and DNA form this kind of bridging-induced phase separation, and you can see here, it's not very easy to see that you don't stop the phase separation here, and that's because in this case the cohesin doesn't really have on this DNA specific interaction. So you wouldn't create loops, but you would create an increased concentration of chromatin, and so the phase separation doesn't have anything to stop it. Okay? So it is still an open thing for the experimentalists to find a neat, if it is correct, a neat case where you have arrested microphase separation. So arrested phase separation, microphase separation in vitro. In vivo, we have the experiments, the nuclear bodies, but it would be nice to find it in a control system. Okay, so this is about the structure. And what I want to know now is try to link it to the function. So these clusters are a little bit like a basic building block of a chromatin structure. I like to think a bit about as an analog of a secondary structure in proteins. Is a bit different, but the idea is that they form uniform clusters along each chain, a little bit like the alpha helices of the beta sheets. So that's a local structure that we are able to understand for the moment. The global, we are still working on it. But the local structure, certainly we think these clusters are very important. So I want to now see whether this structure is just happening in 3D due to this bridging-induced phase separation, or it has functional relevance. And I'm going to do that with computer simulation first. So when you think about functions, one concept is that of gene regulatory networks. So these are typically very complex, and the topology, which is now quite well studied, is basically a question which is now well studied, the topology of these networks. In many cases, these networks are small-walled, or in biological literature, they are called omnigenic. So this means that there is very many interactions. So a small-walled network you can get from any bit, so any node to another node with very small number of steps. So these are, a lot of times it's very difficult to actually construct this network. So the one on the left is a speculation from the biological paper. The one on the right is a pluripotency network, which is indeed omnigenic. So if you touch one node, you will affect every other node, essentially, or any node will depend on every other node, which is a little bit annoying for a modeler, because you need n times n of these if you want to model a network over these. Or many parameters. So the common idea is that interactions are typically regulated biochemically. That's what you can see here. Okay? And of course, this is very important, but what I'm going to show you is that even structure, so even this biophysical interaction and indeed this cluster formation, has some consequences for the biological networks that would come about. So how can we demonstrate this? So we are going to start with a very simple model, exactly the same as we have seen actually, but we just make one key more assumption. So here we have a string of 1,000 bits, which is a toy model for 3 megabase pairs, so 3 million base pair of chromatin. Each bit is 3,000 base pairs in this mapping to chromatin. And we have around 40 here highly sticky bits. And then we make a simulation where we consider a number of proteins, which are complex, which is a little bit less, in this case 4 times less than the number of sticky bits. So the sticky bits will compete for the proteins which are in the sample, okay? So now these are complexes of transcription factor and polymerases. So when they bind here, we're going to say that that bit is transcribed. So that would be a transcription unit in this model. Transcription unit means that it's the beginning of a gene, for instance, a promoter, or it could be an enhancer. So if you know about that, these are regulatory elements. So anyway, the model is very simple. This is our protein, multivalent binder. This is our transcription unit. Here the transcription unit is off because it's got nothing close by. Here the transcription unit is on. Okay, 0, 1, so according to whether physically there is contact between the unit and the bridge or the protein. So then we can simulate do many runs, and for each simulation we can track of 1 and 0. So we can do, for instance, the average and see for each unit, so for each one of those 40 bits, is this on or is this off? Okay, so we can link in a very straightforward way, simple-minded, I would say, the structure to the transcription, which is our function. Okay, so if you think about this as a one-dimensional system, okay, suppose that there is no 3D. The interaction between this red sphere and the pink transcription unit is always the same. So by Boltzmann weight, I need to have that the transcription probability, just the probability of binding, has to be the same for all these bits, right? Whereas if you do the 3D, you see that there is a change about an order of magnitude. Some of the transcription units are always on, and some of the transcription units are always off. So here is an average of 1,000 of simulations, so the error will be very small. So there is indeed this big change. There are some high activity units and some medium and low activity units. So this is a little bit like having a very high variability in the gene expression. Okay, so even though you don't control the interaction, you control, for instance, the positioning of the, so all what this has to do is that this is your transcription probability simulated like this, and this is the number of bits to your closest transcription unit, so to your closest peak bit. If you have a lot of friends nearby, you will be likely to form a cluster. Okay, that's the idea. If you are likely to form a cluster, the transcription probability will be very much higher because it is much more likely that you will suck the proteins from the soluble pool. Okay, and indeed you can do a bit of polymer physics. So this transcription probability decays like L to the -6. So if you've got a neighboring transcription unit, so if a promoter has another neighboring promoter in this model, it will be upregulated. Okay, so one thing that this model tells you is that if you think about clustering as emergent properties, that they shape the transcriptional pattern if you choose to assume that the transcription probability is linked to the binding of a protein to its promoter or enhancer. So another thing that you can get for free is that you can have high variability. So instead of having different cells, so experimentalists have different cells, we can have different simulation. So even if one gene or one transcription unit is very highly active, in a simulation it will be inactive. And so in this case, for instance, this is a histograms for a transcription unit which is very highly active, but you see that it's got a long tail. So this is, if you want, because the system is underlying very slow dynamics or glassy dynamics, and that's why if one cell, if it doesn't turn on, it will stay off for a very long time. And this is again in line with what you see experimentally, although in a very qualitative way. So by this model, you can have a variation over the chromatin, so over the genome, and also variation over cells, so over a population of cells, you have a lot of stochasticity. So I'll also tell you these things that I find quite interesting again in the toy model, and then I'll briefly tell you how you can go to a realistic model. So this calculation has been done by putting together many simulations, and you've got this binding bit index is the index of the TU, so the pink bit. I had 40 pink bits, so there is 40 pixels here and 40 pixels here. Okay, so when you see, so here I'm looking at a different simulation and I'm asking myself, when this transcription unit or gene is higher than the average, what happens to this one? If you see red, it means that they are co-regulated. So if in a simulation 3 is up, 4 is up. If 3 is down, 4 is down, of course, statistically. Okay? Whereas if you see blue, it means that there is anti-regulation. So then you can do a statistical test and ask when this, in this case, is a Pearson correlation, which is shown here, is statistically significant. And you can then use this two-dimensional matrix to draw a network. So the network that you come up with is something like this. Okay, so here you have your 40 bits, and you can look at when, just due to this spatial structure which arises, essentially, you have a correlation between transcription units. You've got a solid line if they are co-regulated, so that's this red clusters, and this means that if things are in the same cluster, they will be co-regulated together, so they will be both up, or both down if the cluster doesn't fall, okay? And then you've got loads of gray regions, and the physical meaning for that is that there is competition. So we have here about, in this toy model, 4 clusters can form, roughly, 1, 2, 3, 4. I don't have proteins for all of them, so if you form cluster number 2, cluster number 4 will not form. And that's the reason why we have the gray correlation. So this structure gives you, the clustering gives you both positive correlation and negative correlation through the competition. And the network which comes about is omnigenic by definition, naturally, without having to put in any biochemist. So that's how you start from, okay? So of course, then biochemical interaction will have to be included, but the structure itself gives you a very detailed, very rich, you would say, or very complicated, if you want, the network. The other interesting thing is that we can sort of have a percolation transition. So hopefully people here are interested in this. And if so the problem here is that the long-range correlation are due to competition. If I increase the number of proteins which bind, I decrease the competition, so the transcription units will not need to compete so much, okay? So basically I saturate them. Suppose that you flood with red proteins, every transcription unit will be transcribed because there is enough for everybody, okay? And that's why you start to lose the correlation here. So if you look at the number of proteins, the percolating fraction of the network, so the fraction of the largest connective component goes down dramatically, and it's extremely of a transition. We don't have more than this, but that's so qualitatively this strongly suggests a transition. And this is also in line with the qualitative idea that if you flood a system, a cell with a transcription factor of a given type, you will make it switch. So whatever is the network of the correlation. So this is perhaps because if you flood with the transcription factors, you simplify a lot the correlation. So you don't have all these omnigenic correlations to worry about. So I'll spend another, I can do 5 minutes or 10 minutes on this. How much do you want? Yeah, so I'll discuss the real chromosomes, which I guess you are interested in, because as a physicist, of course, we like toy models, but it's also good to see how they behave with respect to real chromosomes. And here we learned a couple of more, so we had some results which I want to tell you about, which we found interesting. So this is coming from our collaborator in biology. So it was called predictive heteromorphic polymer. They just put highly predictive and then hip hop page that's become. So it's nice and more. Hop or pop was not there. So the hip hop model has 3 main ingredients. I will just discuss 2 now. Okay, so one I discussed in detail, and that's the bridging-induced phase separation. So I got these proteins which are active and they are multivalent. So these are, so now there is a little bit of biological jargon. So the important proteins here are the orange, that's the active. These are inactive, so they're not so important for the story, but they're included. You need to have all the details, you know, that's what that. And these models is built on the basis of bioinformatics data. Please keep in mind that we do not have fitting parameters. Okay, so the only parameter that you could say is fitting is the affinity for the protein and its transcription unit. But it is provided that it's strong enough to form clusters, it doesn't change a lot. So this ATAC is an assay which is similar to DNA's hypersensitivity. It tells you where the accessible region of the DNA are. So basically it tells you where the promoter and the enhancer are. So we're lucky enough that we know the position of these ATAC sites. These acetylated regions are open chromatin, so that's the third component of the model that I want to describe. The interaction between the protein and the ATAC will lead to the formation of transcription factories in this model, okay, in the hip hop as well. So this is the first ingredient. The second ingredient we put, because so there is a very influential paper, model actually by the Mills group originally, which says that there are motors working on DNA. Not everybody agrees on the nature of the motor, but it could be SMC-like proteins. In this case, it's a cohesin. Cohesin is a protein which binds chromatin quite a lot. And the idea is that when it binds, it forms this loop, so it creates a little hook here, and then extrudes a long loop until it finds some stop signals which are CTCF proteins, so don't worry about them. But these are motors which work on the DNA. So we had different back and forth models on this kind. So we had some, there was some controversy on this model, but for the sake of the hip hop, we are using the Mirny version, so the one which has active loop extrusion, as we call it, okay? So these are the two main ingredients of the hip hop model. I don't show it, but it does the contact map. People now look at contact maps, so the structures are very good agreement with experiments, and I'll tell you two take-home messages on the hip hop. The first thing is that we can predict, like we did before, in a very stupid way, on-off, so whether the protein is close to the transcription unit, the transcription probability, so the transcriptional activity of every bit of human genome, if you want. It can be genes, but it can be any DNA, so the DNA doesn't need to be a gene to be transcribed. So if you look at experiments and simulations, we get a decent agreement. So the correlation is about 0.6 at the moment. And again, there is no fitting. It's just looking at where the transcription units, so the ATAC sites are the human chromosome. So it's quite impressive that given that you don't have the parameters apart from this affinity, if you want. You could also think about the looping, so the extrusion velocity, but that actually doesn't really matter. So this correlates more significantly than previous simplified model. And one thing that we can see is that the nice thing about this, so we have built, so unfortunately it's on the other line, but it's so people on the Zoom will be able to see it. So there is a data set called 3D Gene in Edinburgh. If you Google it, we have the gene structures for all the human genome. So thanks courtesy of Michaels, who has spent 6 months simulating it. So this is for lymphoblastoid cells. So because we have a validation, so the transcription prediction is relatively similar. Okay, the correlation is 0.6, is not perfect, but it correlates quite well with the experiments. We can then hope to mine that data set and see something which is actually interesting for experimentalists. For instance, here we can correlate the simulating transcription rate, which we know correlates with the gene expression, with a number of partners. So the idea is that clusters, so if you have inactive genes, it will often be clustered like this. Not always, but often. Okay, so there is a correlation. The last thing that I would like to say is what about other components of the model? So because we've always been discussing about this bridging-induced phase separation. Another output of the model is that you can look for each transcription unit, for each gene. You can look at the activity in each of your simulations, so like each of your cells, and you can build a histogram. So you have the mean, which is what we have been looking at, which correlates with the clustering and the bridging-induced phase separation, the secondary structure of chromatin. And we also have the variance that you can use as a proxy for transcriptional noise. Okay? So how noisy is a gene? And by mining the data, so this is the correlation that we looked at, so the transcriptional noise here correlates with the occupancy of cohesin. So this is an example where we didn't really have any idea what the transcriptional noise was given to, and we just tried to correlate with 50 observables. Okay, so, and this one is, so it's a weak correlation, but simulations are such that you can make it statistically significant if you do enough simulation. So this gave us the idea that the noise might be linked to this transcription, to this loop extrusion, because cohesin is the motor which creates the loops. And actually there has been a series of experiments which have tried to knock down cohesin, and they found very little effect on the activity, okay? So the idea that we have is that the activity is actually more to be it has more to do with the clustering, and the extrusion changes a lot the noise, because if you have a lot of extrusion in a gene, you will form in different cells different kind of loops, okay? And this will increase the noise. And indeed, if we simulate cohesin removal, this is the mean transcription. This is the mean transcription when you simulate the degradation, so you don't have loop extrusion anymore. There is no change in the transcriptional activity, whereas the noise goes down. So this is an example where at least in simulation we can use some kind of correlation analysis of our database, and we could predict mechanistically what is the effect of a component to the model. Okay? So I'll just briefly conclude. I told you that multivalent chromatin binding leads to microphase separation in these clusters, which are key in transcription factories. They are very much linked to the transcription activity. So if you tend to form clusters, this will make, so if a promoter is in a region which tends to form clusters, it will likely be highly transcribed. Okay? So transcription is determined or correlates a lot with bridge-induced phase separation, and transcriptional noise might be linked to another important process. It's a topic which is described only briefly, which is known as this loop extrusion, which has made quite a big impact in the field in the last few years. I'm finished. Thank you very much.
I mean, that was very nice.
Okay, thanks.
I think you explained a little bit, but I didn't totally understand what prevents the coarsening for the microphase.
Oh, yeah.
I was wondering if it was related to surface phenomena and pre-wetting transitions, perhaps.
So I don't know how. I'm sure there are other ways to see it. So we had a little theory where, if you can think about this, so what we can formalize is that the cost of a loop is, say, C kBT or 10 kBT, let's say. The creation of two loops, you know, this work by Duplantier, for instance, a polymer network, won't be 20, but would be a little bit more. So this C exponent will depend on the topology of the network. So if you use those exponents, you find that you have if you put more and more stuff in a cluster, the energetic gain is the same, but the entropic cost is superlinear. That's one way to put it. Certainly there is kinetic effects. And the one that you suggest on, I think there might be, you might be able to see it as an excluded volume interface. We haven't got a theory on that, but it would be nice to, I think if it is seen also experimentally, it would be nice to work a bit more on that. Yeah. Another thing that we're thinking about is the protein-protein interaction and how this affects, but this is because the biologists are also very interested in this.
Questions?
And
I'm curious whether your model gives some insight into cell states and cell state transitions.
Well, that's an interesting point. So, okay, so for cell state, the model that we have been thinking about it. So the model that we have now is, so the hip hop is predicated on a set of histone modifications. So the simplest way we could answer that is if we had the attack sites which are open and the acetylation for different stages of the cell cycle, we could predict the transcriptional output and the structure. Okay. To go beyond that, you would have to have a model which dynamically changes the bits, essentially, which we have thought about this a bit. And it becomes more complicated. So I don't know. I think the simplest way would be anyway to And this is interesting, for instance, for sure, in our case, because the cell cycle will affect, since we are trying to measure this thing, the noise. So we do have evidence that the cell cycle at the experimental level, though. Yeah, so it would be nice to.
Really interesting talk. Is there enough known about human chromatin to be able to say whether this occurs within chromosome territories or between chromosome territories?
Yeah, I would say
whether it affects the elasticity.
Oh, yeah. I think this is an important point. So it will occur if both within and without, in my view. So between within and between, inter and intra. Yeah, and that will depend. Probably this will depend on the volumes of the nucleus, I would say.
It's a biological question.
Sorry?
It's really a biological question, whether you need localized networks that are different chromosomes.
So you are saying it is a biological question? Or?
That's what
it
all comes down to biology.
Yeah, so the biology that we work with are very interesting. So the chromosomes are rugby balls, so a little bit, so they have quite a lot of surface area. So they think there is a lot of this intra. From the point of view of the models that I told you about, this could have all happened in a dilute system, actually. But it doesn't, and if we simulate the real nuclear volume, we do have intra and intra, and there is not, and I think this would be, there is not really a quantitative final assay biologically. So they know that there is a lot of intra-clusters, not clusters, but the intra-contacts and intercontacts. But if you look at high C, for instance, they have a problem with the normalization. So they usually don't like it when there is a lot of intercontacts. So they have to improve a bit the assay, I would say. But I think it's an important question, yeah.
And then during cell division, these all fall apart and they have to come back together.
So these clusters would fall apart in cell division. Actually, yeah, unless you are heretical. Some people say that there are a little bit of transcription in mitosis, but let's not get into that. They usually fall apart because there are other marks that the ATAC sites are lost, essentially. It's an important question to think about the transition between the two.
Oh, there's an online question.
From Budo, I can read it out. You mentioned protein-induced phase separation. What about the laminin-induced phase separation?
So laminin-induced phase separation, so what I know is that the lamin is normally associated with the envelope. So what I know about the lamin is that it usually is a way to make a fiber inactive, so go to the interface. We did some model with the surface. So if you want to put the lamin in the game, you need to think about a heterochromatin. So certainly heterochromatin will phase separate in a similar way. So there will be inactive regions as well as active regions. Perhaps that's what Budo is meaning, but also if you want to really understand the lamin, you probably have to model the surface of the nucleus, which is interesting.
That's it.
Okay.
Okay. Oh, there's another one?
Budo saying thank you.
Thank you.
Okay. Thanks very much.
Thank you.
Okay.